summer. The benefit here of the institution is that the access to patient material, and not only at a one moment in time, but that we're able to follow patients over a longer period. Now, there is a bias here in part of the center towards pancreatic cancer. And so what I'm going to share with you is, in the context of pancreatic cancer, different immune cell population that are hiding in the tumor microenvironment, and we examined where they are, what they do, what they could do, and what may happen if they go back where they're supposed to be. So a more recent analysis of Guide to Cancer Immunotherapy from T-cell basic science to clinical practice is essentially you have at this table of immunotherapy a number of different instruments. An old horse that is sometimes revived are tumor-infiltrating lymphocytes. At least it's now an approved product for patients with melanoma. Cervical cancer will follow. CAR T cells, epigenetically changed T cells, tumor-infiltrating lymphocytes, bispecific antibodies, you name it. All of them the challenge is that it's a great product, it's an intelligent product, but maybe the point that is really missing in this context is where do they go to? They go to the tumor microenvironment, and this may decide if these therapies are successful or not. So if you look at a very, very high bird perspective at the tumor microenvironment, so there's a lot of stuff going on. You heard it already, and you are going to hear it. One insight from Nicholas Restivo, even that in the context of necrosis, high potassium has a huge impact on the nature of T cells. So high potassium leads to preferential expansion of T cell precursors defined by CD45 array CCR7. So there is essentially, it's a double-edged sword. So in the valley of death, where tumor cells die, the high potassium associated with tumor death leads to long-lived precursor cells that are pretty tough in order to survive. There is low pH, there's lactate, who has a lot of impact on T cell functions, particularly also IL-17 production, glucose metabolism, and so on. And of course, IL-17 production in the tumor environment. And this is not only true for immune cells that produce it. 95% of pancreatic cancer cells have a KRAS mutation, and certain KRAS mutations lead to the fact that tumor cells like a G12S mutation, that tumor cells make IL-17. So there's a lot of things going on at the tumor site as well as at the tumor microenvironment. So there's an imprint. So what I'm going to share with you today is where we're looking at T cells that we get out from this tumor microenvironment. But I think what is going to be more challenging is if these cells are supposed to go back, because it's the same thing here. It's high potassium, low glucose, low oxygen, high lactate, low pH. So even if you have amazing cells, if you have a great product, they need to be changed, or we need to be better than nature in order to do something therapeutically. There was an excellent review regarding pancreatic cancer, what can be done, what are the challenges. One of them was cholecystokinin and the effect of tumorigenesis. There was a lot discussed here and in other places regarding tumor-associated fibroblasts and the plasticity, and there appears to be a certain group, myofibroblasts, that are associated with chemoresistance, but also to resistance to immunotherapy. And the interesting point is essentially to look at the local recurrence, what is different, as well as distant metastasis. We cannot answer here everything, but I'm going to share with you essentially here what's in the tumor microenvironment and what is the landscape. Where do we get these T cells from, and what could we do in order to achieve, to overcome these structures that are so adverse? Now, if we look at a tumor microenvironment and we look at different metastases, this is very clear. It's not only one size fits all. It's like a movie. So if we look at metastasis at one point, we may see this image, or if we look at a distance. So there's spatio-temporal differences in metastatic lesions, or as well as within the tumor, particularly in pancreatic cancer, an entire landscape of different collagens. Some of them are even more immunosuppressive or facilitating tumorigenicity. So it's important, where do we get, in which area of the tumor microenvironment are we looking at for these antigen-specific T cells? And that's a slide that's really important and close to my heart, because this is done from pathology here routinely. So it is not in a research context. This is done in a quality-controlled fashion, and it has an impact. So we know in the tumor, and this ends up as a clinical report. So we know about the T cell infiltrate. So this can be analyzed, digitalized, in order to see what's going on to CD4, CD8, CD68. If class 1 and class 2 is there, common tumor antigens, and then in parallel sections, so we know how the landscape looks like. We're able to look at the T cell receptor repertoire and do DNA exome sequencing, and not only at the DNA level, but also we're able to see if these genes are really being expressed. Now, this is a lung metastasis from a patient with pancreatic cancer, and it's very clear this is CD68 staining. So depending where we run our spatial transcriptomics, or we get our T cells out, if we get them out, we will get a totally different image as compared to here. So this is that's a common theme that we discussed in different areas, that the truth is not only a representative area of the tumor, but the entire tumor. Because what is representative is a rather subjective measurement, depending what kind of filter we use, scientific filter or clinical filter, to look what is relevant. So it may be, for some questions, more important to look for a fishing expedition, to look the entire platform, the entire landscape of the tumor. Now, this is how it looks in reality. If a tumor is being resected, then under the guidance of a pathologist, the tumor is opened, then a part is being removed, and it always ends up here with a parallel section for experimental analysis and mRNA spatial transcriptomics. So if we look what kind of cells are in these specimens, we don't just mash it up like a bouillabaisse and crow something out. It goes based on anatomical zones. Desmoplastic reaction 2 millimeter away from the vessels and so on. So there's a localization, the different zones of the tumor. And I'll come back to this. And this is going to be guided and scaled by H&E, and also by much more by smarter immunofluorescence staining. Why is it important? Because if you look at the tumor, and this is, you heard this many times, you're going to listen to this also in future topics here, is that you have within the tumor some MHC class II expression on tumor cells, not only on tumor-infiltrating macrophages, and there are tumor centers that are class II negative. Tertiary lymphoid structures are in pancreatic cancer, as well as in non-small cell lung cancer, usually a marker of good prognosis and response in general. So we don't see a lot of loss of MHC class I, at least not in the tumors that are being resected. And again, these are the good ones. These are the 20% who are resectable, 80% are not. There are CD4 and CD8 cells. There are B cells, CD20 positive cells around the tumor, and there is usually an infiltrate at the tumor rim. So what cells are we looking at? The cells in the tumor, at the tertiary lymphoid structure, or at the tumor rim. This is a very crude way to look at it, but the point here I wanted to make, if you look at this resected liver, and I met last evening when we had this meet the speaker thing, somebody said, well, the necrosis is really important. Necrosis is not only dead cells. And this is exactly what we found here also. It contains a lot of potassium, but also long-lived memory T cells. If you just look at this CD8, so we have CD8 here, 20%, it increases as far we get away from the tumor. And if you look for KRAS reactivity based by gamma-interferon production, so we have KRAS reactivity here and here, and mesothelin, a commonly shared non-mutant target, here. So essentially this shows that localization is important, not only at the tumor and microenvironment, but also macroscopically. Now, what helps us in order to really to go deeper is either to go for individual cells with laser microdissection, or essentially to discard areas that may be not beneficial from a technical standpoint, to remove desmoplastic reaction, to remove certain collagens or cells that may inhibit T-cell growth. It may also help us to enrich certain areas to make a T-cell product, or it may help us for developmental research to go for T-cell receptors or T-cells that sit very close to the tumor. Of particular interest are the CD4, CD8 double positive, highly activated T-cells that are cytotoxic. What you see here in the immunoassaying is essentially you have a quite beautiful nest here of T-cells and a surrounding gamma interferon positive cells. So they're not T-cells. They may be ILCs. There may be NK cells. So, and these T cells then make a different quality of the cytokines. So the tumor microenvironment, so the gamma interferon is not only provided by T cells, and if we look for IL-7, which is a very interesting cytokine, it's one of the most potent TGF-beta antagonists. It's a T cell non-die factor. It helps T cells in a negative way in autoimmune responses to live on, but it helps also T cells in tumor to hang on and to be active. And they can be expressed by dendritic cells, by certain types of macrophages, but also by tumor cells. So we have the IL-17 here, and very close you have this T-cell lymphoid structure composed of T cells. So that's an interesting area to go for and go fishing for tumor-reactive T cells. What you see here is each line here is one TIL, tumor-infiltrating lymphocytes, from one individual patient. And these are not private targets. We did this for a good number of individuals. These are commonly shared targets. Mesothelin, MUC4, MUC16, different KRAS mutations. What you see here is gamma interferon production, and what you see that at least one epitope is usually recognized in these T cells that can be very robustly expanded from pancreatic cancer lesions. Now, not so fast, because we see it, but we obtain this if we go by zone. Now, let's just entangle that here. What you see here, not very clearly, is essentially a collection of different commonly shared tumor targets. So you have different mesothelin peptide, KRAS. It's essentially a test battery. And in zone number 1, there is no gamma. The T cells grew, they're beautiful T cells, they have beautiful phenotype, they may do all kind of things. There's no gamma interferon production and no IL-17. Zone number 2, well, there are 2 targets being recognized, and this is measured as gamma interferon per 10 to the 4th cells. But quite some IL-17, not so good, because IL-17 may provide pro-inflammatory signals and promote tumorigenesis in this context. Only at zone 3, we had no IL-17 production, and we had a very broad recognition of this tumor-associated antigens and gamma interferon production. If you look at the T-cell receptor analysis, that is routinely done. And what you see is here the alpha and beta chain. Each color is one individual V beta family or V alpha family, and the size of that ladder code is the abundance, the relative frequency of the T-cell receptor CDR3 region that contacts the target. So you see you have a pretty diverse TIL repertoire here, and that is derived here from the tumor. If we look for private mutations, this was, I think to my knowledge, the first time that we are able to find a target that is a mutant protein associated with the mitochondrial membrane, which was recognized in the context of HLA-DP402. And the autologous tumor was recognized. This is gamma interferon production. not blocked by class 1, but blocked by anti-DP. So essentially it works for commonly shared targets, but also for, it recognizes tumor-associated antigen. Now, this is essentially, if we look for this really beautiful cell paper that is sometimes forgotten in pancreatic cancer is here. This is full, this doesn't really, well, here it is. It's full of IL-17 and TGF-beta. So that's the tumor microenvironment we are looking at. And if we, and particularly anti-KRAS responses, and this recapitulates what I just showed you in the slide from zone number 2, it's particularly the KRAS mutations that elicit a very strong IL-17 production, but not gamma interferon. Now, how can we really defragment it and get more information? So if we slice a tumor, we can show the diversity based on DNA exome sequencing. We can put all the cells in the air and do single cell sequencing. But primarily a smart approach is also to do spatial transcriptomics. What is the difference and what is the relationship of these cells to each other? In order to do it and also to show not only also function, is we need to grow them, particularly if we go for low frequency events. And this is possible to grow up cells to billions, and then we can really go to low frequency events like mite cells, I'm going to share with you, or gamma delta T cells that are part of a mixed bag, and they're usually overloaded. So what we did is what we do in these cells, we look for proliferation, the nature of the recognition, and spatial transcriptomics, and disentangle that. The important message is here, using this expansion procedure, and these are T cells that are expanded from patients with pancreatic cancer, that we see in 7 out of 7 induction of 2 genes, CXCL10, which is associated with invasiveness into the tumor and has an anti-angiogenic effect. And this could also be confirmed by protein expression. So that's a good thing, so that these cells, at least where we expand, are able to go back into the tumor and able to make their way. This is Eric de Souza's Benfica fan approach to look at the T-cell receptor repertoire, because you have so many indexes. Everybody has his favorite index. And his way was to look, if Ronaldo tries to get in the draw, people look the left leg and the right leg and what he do and how he interacts. So at different parameters, and he was able to put all these parameters essentially in one graph. And this helps us really to appreciate, let me see what's going on here. So we look at the tumor, we look at the alpha chain, the beta chain, the gamma and delta chain, and here you have the different indices. So we're able really to measure from a very deep but also high perspective, what is the repertoire? For instance, the Gini index looks at the inequality. What is the diversity in the tumor? The Shannon index, how many T cells are really there? It looks at abundance, and if you pick randomly something, that it's not, that's not the identical T cell receptor. And we see a similar repertoire in the TIL. So how does it look like when we expand these cells from the tumor? So there are some examples. So we see pretty much that the diversity, and again, these are these beautiful plots that that we can generate if you go online. It's TCR cloud, and what you grasp here is actually you have a very broad repertoire in the tumor. And also reflected, except this patient 1313, the laser doesn't work very, but you see it anyway. So you have a pretty broad repertoire in these T cells that are being expanded, and very important. So these are individual patients. This is gamma interferon production directed against the autologous tumor, and some of them are HLA-ABC blockable, so they're class 1 restricted. And some of them are HLA-DR restricted, blockable by an anti-MHC class 2 antibody. And we know the signature. We know exactly when we sequence the tumor, what kind of mutations do they recognize. So we are able to have a deep dive of these cells. Now, if we look essentially what's going on in spatial transcriptomics, we see a lot of CD4, and there are only a number of CD8s in certain areas. The CD4 is, that's a false positive in a way, because it's not only CD4s that are active that express CD4, it's also tumor-infiltrating macrophages, CD68 positive T cells that do that. So, so we were able, this is Jessica from our lab actually, she took the endeavor and said, well, is there something else in these cells? And we had billions to look at, and if we look at mite cells are very, they came up 10, 15 years ago. They recognize, in the context, vitamin derivatives. This is how the whole field started in the context of MR1, a commonly shared target. And there were two papers, one for 2 years ago, I think it was published in Nature, that it was a specific T cell receptor against MR1 in epithelial cancer. The target has not yet been debunked. And we see this also infiltrating here. In context, if we, and everything is in a way in the corner. If we look at gamma delta T cells, they're the only one that we see throughout these panels that really go into the tumor. So there's something particularly interesting regarding these cells. And if we look here in our patients in the peripheral blood versus tumor-infiltrating lymphocytes, at different markers, we do see that they have a totally different phenotype. They are more activated, they're more antigen experienced, and they are able to infiltrate into the tissue. Are they active? Yes, they are. These are autologous tumor cells with positive low sorted mite cells. This can be blocked with MR1, an antibody, not blocked with the isotype. And the different colors is it was harvested at day 3 and at day 7. So these mite cells recognize the autologous tumor. Is it a shared antigen? Yes, it is, because if you use an allogeneic tumor and knock down the MR1 with siRNA, it's not recognized anymore. So there is a certain subset of mite cells that are part of the TIL, they are infiltrating in the tumor, they're not, that are tumor specific and not only reacting against vitamin metabolites. I think I can spare that. There's something very particular, and this is new, and this was really to our surprise. What you see is here in gray are not mite receptors in the tumor or in the TIL. So you see almost no yellow and no blue because the frequency was so low. Now, the V alpha 7.2, which is the receptor of these T cells, these are the ones, there are hundreds now that have been reported. They're just a few. And this was really surprising to us that the diversity, we thought they have boring T cell receptor, they're all more or less the same. It's not true. There is quite a diversity in this T cell subset, and they have private T cell receptors. In the poster, you will see that only a few that are shared. So there is quite most likely an antigen-driven expansion of these cells in situ. And these are this beautiful bars. So this is an index that you would like to see. There's a high Gini index, essentially. There is some inequality, there's some richness in it, and with a low Shannon index, except for one individual. So there appears to be much more going on in these tumor lesions. The gamma delta T cells, they're the only ones that go really into tumor. If we get them out, and in blue is the delta 1. And they have been negatively sorted up to almost 90% purity. And that's the immunohistology. You have almost the impression that they kind of sit and almost phagocytose a tumor cell. And then we're able to see that they are able to recognize the autologous tumor. Most of them are blockable by CD1D. Again, these are purified. And this gamma delta T cell, and about 50% of TILs that we isolate recognize the autologous tumor. That's the bad news. There's a lot of stuff going on that is not very helpful. that's in situ TGF-beta expression, TGF-beta 1, TGF-beta 2. This will put a stop to everything that you put in a tumor or infuse systemically or locally. And how can we change it? And I would like to end with that on a more positive note. One way is essentially pro-tumorigenic is IL-39, IL-35 produced by B cells. You go back to the anti-tumorigenic IL-12, which is quite toxic in a way. Late 90s, beginning 20 years ago, a number of IL-12 trials. But there is a regained interest. How can IL-12 reshape the tumor microenvironment? And if we go for single-cell secretome analysis that we perform in these cells, you see here in blue or in control in this kind of dark red, and then if the cells are activated with anti-CD3, they move over here and have a different secretome. What you do in a secretome is you look at 35 individual cytokines on the single cell level at the same time. If you add IL-12, they end up somewhere else. And essentially you have a different quality, so you are able to give the T-cell receptor sequencing signal and have signaling through the IL-12 receptors profoundly changes the secretion of the production of cytokines. Is it lapolar? Does it have any impact? Yes, it does, because if you look at private mutations, so I walk it because it doesn't work anymore. If you look at gamma-informed production, in each of these different graphs is a different concentration of IL-12. So the more we increase the IL-12 production, the priming of these T cells, the more targets we identify. And lastly, how do we get in a tumor? How do we to get in this bad environment? One way is essentially to have access and create a tertiary lymphoid structure. And one of the hallmarks is CXCL13 that is produced by follicular dendritic cells, particular subset of T helper cells. That's not a tumor. That's an organ-specific autoimmune response with a hallmark of a lymph node in an organ. And this is, if we look here in our tumors, we have CXCL13, you see it here in the corner, CD3 epsilon, and if we overlay it, essentially they're together. So we need CXCL13, which in part is also a double-edged sword, but without CXCL13, there are no tertiary lymphoid structure. So the take-home message is what we see in this context. We have alpha-beta T cells. If we get them out of the tumor microenvironment, which is localization, localization, localization. So we have T cells that have a recognition pattern, but also totally imprinted different cytokine production. Mite cells, gamma delta C cells have their own dynamic. It appears to be a strong localization and imprint. And this, of course, helps us to think about how we can manipulate the microenvironment. And I just shared with you very superficially two different examples, CXCL13, or the pre-priming of immune cells to give a signal that they make more gamma interferon and really are able to expand cells that have a better epitope focus. Now, when this came out in the New England Journal, we were very happy because what people described as the pathway that we have to have a renaissance of looking in situ. We felt that we can tick the boxes. We do the spatial transcriptomics and we look really at the individual responses, and I think what we have here is only the surface, but what we really need to do is to have a single cell in situ sequencing for the T-cell receptor repertoire, which is a paper that came out last week from Leo in Immunity, in order to marry this with the tumor microenvironment and really to have a better understanding of the heterogeneity, but also the quality and the nature, and how we are able to be maybe better than nature impact to reprogram that sometimes fertile soil or hostile microenvironment. So to summarize, we have a seamless interaction. This made it only possible to biopsy in the surgery. TIL and GI cancers are diverse. There is a hallmark of the Th1, Th17 pattern. This is very much preserved, and the TALER therapy needs to address the patient's immunophenotype So it's not only different from patient to patient, but also in the localization. And this is one of the, that's the crew essentially who made the first billion T cells. And this would have not been possible without our surgeons, Jill Gonzalez, Professor Büchler, the oncologists, the pathologists, and then we have all the guys here that you're going to meet here at the posters and who work together, but each one of them has its own baby. The mite cells and the gamma delta T cells. I hope I was able to give you a short vision of what we have been doing and the challenges that are ahead. Thank you.